Dear participants, good morning. On behalf of the organizing committee, I welcome all of you in the 12th technical session of Total Online FDP on Data Sciences. The resource person for this session is Dr. Khalid Raza, sir. Dr. Khalid Raza is an assistant professor at the Department of Computer Science, Jamia Millia Islamia, New Delhi. Dr. Raza has been honored with ICCR Chair Visiting Professor by Indian Council for Cultural Relations, Ministry of Foreign Affairs, Government of India, and deputed at the Faculty of Computer and Information Sciences and Shams University, Cairo, Egypt. He has over 10 years of teaching and research experience in the field of translation bioinformatics and computational intelligence applications in healthcare. He has contributed over 70 research articles in reputed journals and edited books. He has authored edited books with reputed publishers like Springer, CRC, and elsewhere. He has reviewed over 150 research articles for reputed journals or conferences in the last 5 years. Dr. Raza has delivered several keynote addresses, invited talks, public lectures, and seminars in national and international conferences, workshops, and chaired technical sessions at various conferences. He has served as the technical program committee. He has served as the technical program committee members of several international conferences and workshops. He has also executed two Indian government-funded research projects. His research interest include his research interest includes computational intelligence method and its application in bioinformatics. We are fortunate enough to have a session with Dr. Khalid Raza, sir. Thank you, sir. It's over to you now, sir. Thank you, sir.
Good morning, everyone. Uh, First of all, I would like to thank you, Dr. Sajid, for introducing me. My today's talk title is Computational and Big Data Challenges in Bioinformatics Research. Here in this lecture, I will talk about some of the big data challenges and some of the computational intensive tasks that we do in bioinformatics. I was told that the audience are from different disciplines, so I will try to keep my lecture very simple, and I will not go in much detail in the algorithm, but and also not will go in much detail in biology. Rather, we will see some of the sources of the big data that is being generated in bioinformatics, and how we are handling such voluminous data, and what are the computational challenges that still are there in the bioinformatics area, and some future prospects that we will see. So the content of my lecture is first I will introduce about some brief overview about the field of bioinformatics, then major branches, then we'll see growing omics based domain. I think you must be knowing about various omics, genomics, transcriptomics, proteomics, metabolomics, and there are so many omics today. So I will just mention about those omics. Then prominent repositories and databases and tools. We will see how the biology is contributing voluminous data from different sources, and we'll also see how these data are publicly available for all of us to analyze these data. So that most of the data produced in the field of bioinformatics, more than 90% of the data is freely accessible for everyone. Then we will see the holistic view of biomedical science research. We will see how bioinformatics is contributing for a better treatment and better healthcare in the field of biomedical sciences. We'll see the big data, the sources of the big data in the field of bioinformatics. Then we will see major computational intensive tasks in the field of bioinformatics that are posing computational challenges before the scientific community. Then we will see big data problems in bioinformatics and future prospects. So that's the content. So I would like to start my lecture with the motivation that is computer science perspective by this Donald Knuth. Donald Knuth, I think you must be knowing it, one of the leading scientists who has worked a lot in the field of computer science. So, being a computer scientist, why somebody is so much curious in the field of biology? I have extracted some of the code from the Donald Knuth speech, which was delivered in 1993, and I'm so much inspired uh, with this quote. Donald Knuth said that biology is so digital and incredibly complicated. I cannot be as confident about the computer science as I can be about biology. And biology easily has 500 years of exciting problems to work on, and it's at that level. So, what Donald Knuth has told in during 1993 about the biology, that biology is like a digital, what we do in computer, the thing which is represented as a 0 and 1, and it's very similar to the gene expression of our body. Sometimes our genes are expressed and sometimes our genes are not expressed, so it's like a digital circuit. So he has compared the biological system with a computer system. The most important thing here he has mentioned is the biology has 500 years of exciting problems to work on. So we have a variety of research problems in the biology. So once you will go inside the biology to understand how the things are done, then you will realize that there is nothing is done in the biology. Still there are lots of scope to work upon. So let's define the term bioinformatics. What is a bioinformatics? It's a very new area. It's not very old and very mature area. Uh, it's uh, it's one of the new area. So bioinformatics is not just computational biology. Sometimes we are adding computational term with a different field of studies like computational biology. Rather, it is more than that. The scientists from different fields, like scientists from engineering, computer science, chemistry, biochemistry, statistics, mathematics, biology, all communities are communities of scientists are contributing something to the bioinformatics. So we can say that the bioinformatics uh, term is not very well defined because because I am from computer science, so I can see bioinformatics from my computer science perspective. Somebody who is from biology and they are working on the bioinformatics, they will see it in a different way, right? So uh, several scientists has has uh, uh, given the definition, but these definitions are not very perfect, right? So one of the quote is from Nancy Lorenzi. uh, she told that bioinformatics is like an amoeba, right? It comes in various shapes and it comes in various sizes. So we cannot actually bind the boundary of the bioinformatics. The bioinformatics has very a kind of umbrella term that can cover beyond the computational biology. So here I'm presenting you some of the formal definition that was given by some of the society or from Oxford Dictionary. Oxford Dictionary defines this term as the science of collecting and organizing complex biological data, such as genetic codes. So this is one of the simple definition, that collecting and analyzing the complex biological data. So basically, the main thing in the biology we can understand with this definition is that we have to play with the data. So we have to collect the data and we have to analyze the data to discover some new knowledge from the data. So what it does. Another definition is from NCBI, that is a more exhaustive definition. It is a field of science in which biology, computer science, and information technology merge into a single discipline. The ultimate goal of the field is to enable discovery of new biological insights as well as to create global perspective from which unifying principle in the biology can be discerned. Right. So this is some of the definition that it is a field of study. It's an interdisciplinary field where all the scientists, come together to solve various biological problems. This is a figure of photo of Margaret Oakley Dehoff. She is known as the mother and the father of bioinformatics. So she has lots of contributions in the field of bioinformatics, and she has contributed a lot before the term bioinformatics was coined. The term bioinformatics was coined in late 1970s, but Margaret Dehoff, she has created an atlas of protein sequence and structure in 1965. So she has, contributed a lot in the field of bioinformatics. That's why she is regarded as the mother and the father of bioinformatics. So still we use, many things that was created by Dehoff. Like she has invented single letter code for amino acid. Earlier we used to use 3-letter code for amino acid. That takes too much computing power and storage also. She has invented single code. Another one, she has created a matrix that is called point accepted mutation matrix. This matrix is used for the sequence alignment, and this matrix is still used for finding the evolutionary relationship between the species, how two species are evolutionary related. So still the PAM matrix is being used. She has, many more contributions. I have not listed here. Now, Pauline Hogway and Ben Hesper, there are two scientists who has coined the term bioinformatics in early 1970s. So the term was coined during 1970s, and the term was coined by Pauline Hogway and Ben Hesper. What are the aims of bioinformatics? The aim of the bioinformatics is to analyze the data. So the data may come from various sources, right? So here in this diagram, I have just mentioned multi-omics data like genomics, transcriptomics, proteomics, metabolomics, and we have many more omics. So we can call it as a multi-omic profile. So multi-omic profile, what is the purpose of all this multi-omics data? The purpose is for a better healthcare. So we can call it as a healthcare analytics. So this data can be used for the healthcare analytics, and that can be combined with other clinical data like electronic health records, clinical information, patient health records. So all this data can be integrated together, and then we can apply some computational biology, bioinformatics, and one of the area that is systems biology. We can all integrate and analyze the data for the purpose of personalized treatment. Right? So I will come on the personalized treatment later on during my lecture. So this is the basic aim of the bioinformatics. In other words, we can say that the bioinformatics is just to support the biological and biomedical research for getting better insight about the biological phenomenon and to get better healthcare treatment. The three major branches of bioinformatics. So how to classify or how to categorize the bioinformatics. So there are several different ways to classify the bioinformatics. So on the basis of the central dogma of molecular biology, where we have a DNA, and DNA is replicated, DNA is transcribed to RNA, and then RNA is translated to protein. So that is a phenomenon that is well known, that is known as central dogma of molecular biology. So on the basis of that central dogma, the major branches are genomics, right? We can say, we can correlate this genomics with DNA. So some data which is related to the DNA and produce with that is called genomics. And then this is messenger RNA, right? That is called transcriptome, and this is about the protein. So there are three different three different branches of the bioinformatics on the basis of the data. So what is the genomics? So basically, genomics is the study of the genes or the sequencing the DNA for the purpose of analyzing uh, the genetic code. Then transcriptomic, it is the study of the messenger RNA, and the ultimate goal of the transcriptomic is the gene expression study. So we try to understand the expression pattern, expression profile of genes with the help of different methodologies that we have. Today, we have a microarray, we have RNA-seq for the study of the whole genome data. Then proteomics. Proteomics is another classification where we try to study about the entire protein of a particular organism. Similarly, beside those three major areas, we have also many more omics like metabolomics. Metabolomics is the study of the entire set of metabolites that we have in the body, and we try to understand those metabolites and their role in the body and in different chemical processes. Similarly, we have chemoinformatics. It is also one of the very growing area where we try to study the natural products and their role in the chemical processes. Now, the view of the past, present, and future of bioinformatics. When we talk about during the 90s, the people were just creating the databases, and those were the primary databases that consist of large experimental data in genomics and proteomics, right? Like if you talk about Human Genome Project was around 1 decade long project, and out of that several databases was created, and those databases was the primary databases. What was the purpose? The purpose was to understand the function and the utility of individual genes and proteins. Presently, uh, mostly we are working on the secondary databases that's consisting of accumulated biological knowledge. So whatever that we have or already have in the primary databases, we are trying to extract some knowledge, some information from those primary databases, and we are storing it as a secondary databases. The purpose of creating the secondary databases is understanding function and utility at the molecular, cellular, and organism level, right? But earlier the scale was very small. The just scale was at the gene level or at the protein level. But presently now we are working at the organism level, either at the molecular, cellular, or organism level. In the future is that a complete computer representation of a cell and the organism. So understanding the basic principle of higher complexity of the biological system. So today we are researchers are trying to create the simulated version of cell artificial organ development using the bioinformatics and other engineering field of studies. So this is just about the data. Like data is omnipresent, data is everywhere, it's present everywhere. Uh, Data is expanding on a magnanimous level every day. You must be seeing that the data is being produced, every day. And a single person, if a single person can assess about himself or herself about the data that he or she produces every day, then you cannot imagine how much we are producing. This rapidly growing data can be accelerated further with the help of IoT technologies that today we have. The IoT also has accelerated the data production. Almost 18 billion gigabyte of data is created every day from various sources. 80 billion gigabyte of data. And out of the total data generated, 10 to 30%, 10 to 30 terabytes of data is contributed by the biological experiment in a day. So among all those data production, the biology is also contributing a lot in the data production. These are some of the prominent repositories, databases, and some of the tools that are widely popular in bioinformatics. I have not listed here. Today we have more than 20,000, we have more than 2,000 databases that comes in the area of bioinformatics. More than 2,000, that you cannot imagine. And there is one of the publisher that is called Nucleic Acid Research, NAR. They usually publish this kind of paper where you have created some kind of databases, maybe primary or secondary databases. But here I have listed some of the very prominent repository like NCBI, MBEL, DDBJ, PubMed, PubChem, PDB, and Uniprot. And we have some of the tools like Scodinger, Autodoc, Gromax, NCBI Blast, right? But if you talk about the databases like NCBI, Imbel, and DDBJ, all these databases also comes with some kind of utilities. It's not just a database, rather it is clubbed with some kind of utility as well. Utility for the purpose of data analytics, right? So basic analysis can be done with the database itself. It's not just a raw database, rather it has some analytical tool, but those analytical tool are very limited, so that and that has been designed for a very general purpose, right? For filtering information, for doing some kind of basic alignment of the sequence. This is the home page of the NCBI. This is the world's largest public repositories of biological and scientific information. This NCBI, it is a National Center for Biotechnology Information. It comes under the NIH. It is one of the project of NIH, National Institute of Health, and it has so much databases. So basically today the NCBI was created in 1988, serving from the last 32 years. So this database is still maintained from 32 years. And every day, on a daily basis, this data is being, modified, and data are uploaded. And the best part is that it's not only, the data is not only uploaded by the NCBI, rather the data can be uploaded onto this platform by any research lab across the world. So it's open for the submission of data. So it is maintaining over 40 integrated databases. It's not just single, rather 40 databases. 3 million visitors per day, and it has 27 terabyte of data is downloaded every day. 27 terabyte of data is downloaded every day. So it's very huge data, which is being downloaded on a daily basis by the researchers across the world. It has a single search engine that is called Interest System. So here in the snapshot, you can see here we have a search system here. So it follows the Interest System. So it's not a very simple search engine, rather it is a very advanced kind of search engine that gives almost accurate information, whatever the query that you are going to write here. And it also supports the various logical operators like and or, and it also supports the search on the basis of fields, whatever field that you wanted to study, whatever the organism you wanted to search. So this search is not very simple, rather it is a very complex and very advanced. Tools and APIs for downloading the customized data. It provides you the third-party APIs as well. So within a program, you can connect with the NCBI and then you can download the data, customized data. It also provides facility to submit, download, and learn. Now here we need to understand the holistic view of the biomedical data sciences. Holistic view means that we have the various multi-omics data and how these are multi-omics data like genomics, proteomics, imaging, health record, wearable devices, how these all are, being merged together. So the confluences of the large scale data, that is connecting all these omics data, healthcare data, biomedical imaging data, and how we are applying the statistics and computer science for its analytics. And there are 4M process framework that is being followed in the biomedical data science analytics, that is measuring, mining, modeling, and manipulating. So, and because these data are big data, that so we need to understand the 5 V's data framework as well. On this slide, I just would like to mention that the human body is a source of a big data. If you will see, we have trillions of cells in a human body, trillions of cells, and each cell has so many further information. So trillions of cells in a human body, we have 3 billion base pairs in the DNA. We have 23 chromosomes, more than 20,000 genes, around 95,000 known proteins that still we know. There are several proteins that still we don't know. And we have several thousands of non-coding RNAs. And in general, on 30x, uh, on a 30x, the NGS, next generation sequencing machine, can produce 200 GB of data per sample, right? So for a human being, suppose I myself wanted to sequence my genome, and on a 30x, the NGS will produce a data of 200 GB per person. So it's a very huge data that is very difficult to analyze today on a traditional machine. 200 GB, per sample. It's very huge for a traditional machine. So how big is data? So in concern to, or in related to the bioinformatics, uh, the term big data is defined as a data that becomes too large and it is highly difficult to handle, process, store, and manage. And the various sources of the data in the field of bioinformatics are biological data sciences, chemical data sciences, and biomedical sciences. So there are three major sciences that are contributing the various data, even from the chemical, protein, chemical structures like drug structure that we have. So we have the PubChem, one of the database known as PubChem, where we have the structure of the almost all the drug compounds and other complexes. So today we have more than 2,000 biological databases, but the real-time analysis of these data is very difficult. So we cannot analyze this data in a real time. Analysis takes lots of time. Sometimes it takes a year, sometimes it takes a month if it is done on a high performance computing. So the data is increasing in the field of bioinformatics, mainly revolving around the next generation sequencing, SMRT technology, and nanopore sequencing technology. Then the data is also coming from genome, proteome data, clinical data, imaging data, various secondary data. So these data need to be stored and analyzed in an efficient way that would help the doctors, clinicians, and other healthcare professionals for making the best decision. Best decision about the treatment, best decision about the surgery. And also we have in the field of bioinformatics, we have several very good projects. Like one of the earlier project was Human Genome Project. Human Genome Project was earlier, it was the largest project that took around 1 decade to complete, but it has produced the data at a very voluminous amount of data, right? And inspired by that Human Genome Project, there was several other projects that was developed, and that has produced very large scale data. One of them is a One of them is a 1,000 genome project or 10,000 genome project. So this 10,000 genome project, it contains almost 260 terabytes of genomic data. 260 terabytes. So it's again very huge data, and that is distributed over 250,000 publicly accessible files. So this data is split into different files, different independent files, and number of files are almost 250,000. And these all 260 terabytes of data is accessible freely to all of us. Next is a very important project was the Cancer Genome Atlas, that is popularly known as TCGA data. So many researchers who are developing some machine learning based model, machine learning based some approach, and they wanted to test their model. So most of them they pick the data from this TCGA because it's a very huge and very good repository. It contains over 20,000 primary cancer and also matched normal samples and spanning over 33 cancer types, and generated around 2.5 petabytes of data. So you have almost 33 different types of cancer data, and not only just the cancer data, you will also get the data of various stages of the cancer. And after the development of the TCGA, there are several other groups has created some analytics platform for analyzing the TCA data. So they have developed very good interface where you can choose a particular cancer and you can do several statistical analysis very easily with the help of those interfaces. But in the background, they are using the TCGA data. Next is the SRA, Sequence Read Archive. This is one of the very important source of NGS data, next generation sequencing data. It has around 250 petabytes of NGS data and alignment information. And on a daily basis, it is being increased. Rapidly, it is growing. So we cannot fix that today it has 250. Suppose I'm presenting it today, it has 250, tomorrow maybe, it may further grow. Next is a gene expression omnibus. It stores the gene expression data. It has around 4.5 million samples. 4.5 million samples. Sample means that one human being or one species, we can say, one organism. That so 4.5 million samples from around 2,900 different organisms and hundreds of diseases. Right. So I have mentioned here the purpose of presenting here on this slide was that I just wanted to show how the bioinformatics and biology is producing the data, and what are the various major sources of the data that we have. Right. But I have mentioned already earlier that we have around 2,000 databases. So I have just mentioned here a few, but you can imagine that all the databases, all the 20,000 2,000 databases must be having, giving you very enormous voluminous data. This is a diagram. I think it must be visible. This is a diagram which has been taken from SRA. This is a real-time diagram that is being provided by the SRA database. SRA Sequence Read Archive. It has around 45 petabases, and you can see from 2009 to today 2020 how the size of the data has grown. So you can see here it's almost vertical growth. The data here you can see the data which is in the blue color. This is the total bases, and the line which is in the yellow color, this is the open access data. So you can see that most of the data is open access data. So yellow line represent the data that you have that is open accessible. So still it is growing on a daily basis. This is a growth of MBEL data, MBEL EBI data. It has around to 200 petabytes of data. You can see here the data of Air Express, EGA, ENA, Pride, PDB, metabolites. But and over the time, it is still in a growing trend. Now come to the data storage models in the bioinformatics. There are various formats for different data storage in the bioinformatics research, and most of the data are stored in an unstructured way because the biological data are not very structured. The data are unstructured, or sometimes we try to make it in the form of a semi-structure. So the characteristics of the good data format is it should be compact, readable, represent all category and numerical features, be local and doesn't get changed. Some of the example are like CSV, comma separated value, TSV, tab separated values, text format, and XML. We have JSON also. JSON is also being used here. ARFF format. For a sequence file format, we have FASTA, FASTQ, SDF, SDF5 format. This is one of the very recent file format to store the data uh, in a distributed way, so that that should be compatible with the Hadoop. Then alignment map file, we have SAM file and BAM file format. Variant call format, we have VCF. Protein structure, we have PDB format, and many more. So there are various ways, but we will see that most of these data format that I have mentioned here, these are either unstructured or semi-structured. Now come to the major computational intensive task in the field of bioinformatics. We have seen that the biological experiments, whether it is next generation sequencing or Human Genome Project or TCGA project or 10,000 Genome Project and many other projects, all those projects has generated enormous data, and most of those data are unstructured in format. And these all databases, these all data which are produced are just dumped into the biological databases. Why I'm calling it as a dumping? Because those are the raw data, and those are the raw data that is not human interpretable. Uh, Even the simple analytics we cannot apply because that data is totally raw data. If you will see our human genome data, like DNA sequence, we cannot interpret the DNA sequence, right? That DNA sequence is written in the form of 4 bases ATGC. So it's just ATGC, the occurrence of these 4 letters. So we cannot interpret those data, right? So we need, before we are going to convert this raw data into some meaningful information, basically we need lots of data pre-processing. So data pre-processing is one of the very important aspects here, where we will try to convert the data or pre-process the data into some of the format that may be not directly interpretable by the human being, but at least that should be interpretable by some analytical tools. So here we will see, because we have a very huge amount of data, and there are many tasks that we perform in the bioinformatics that requires the huge computing power. So one of the computational challenges in the field of bioinformatics is the next generation sequencing data analysis. If we talk about the next generation sequencing data analysis, here we have a very large amount of data. We have a very large amount of data. If we'll try to consider a particular data, single sample, generally we work on thousands of samples, but I'm talking about the single sample. The single sample can be minimum 2 GB to maybe 200 GB, a single sample. And generally, I have already mentioned you that there are lots of data pre-processing. So the data pre-processing takes lots of time. Here we can see that the raw data may be, between 5 to 200 GB per samples. Here I'm talking about the Homo sapien data. If you'll take some different data of a smaller organism, the data size may be smaller. But if you talk about the human data, so Homo sapien data will be within 5 to 200 GB per sample. That depends on the depth coverage. So how much coverage that you are considering, more and more coverage that you are going to consider, or more and more resolution that you are going to cover, the more and more larger data it can produce. So a single sample can produce this much of data. So that is the raw data, and the raw data is just in the form of like this, like it will have a sequence data, ATGC, so occurrence of the just 4 base pairs. And here we can see this is the short reads. So in single sample, there may be around uh, it may be around 400 million short reads. So we have a 400 millions of the short reads. So we can say that these all are stored into a single file, right? And that is known as FASTQ file. So, and that is a very huge file that we cannot open even the file on a normal computer. So once this is the working pipeline of the NGS. We have the raw reads, then we go for a quality check, and after that we go for a mapping algorithm. So the mapping takes lots of time. It is very computational intensive task, where what we have to do, we have to map the entire short reads to some reference genome. So here we will be having some reference genome, and we have to map the entire sequence short reads to the reference genome. And how much we have? We have almost 400 million short reads. So what the algorithm will do? Algorithm will pick one short read, and then try to align to the reference genome. And reference genome is also very, very long. It has the size of the reference genome is human genome is a 3.1 billion base pairs. Right. So 3.1 billion base pairs means that if you will try to write the entire genome of a human in the form of a printed book, it will be around 3,000 books. So it's not very small data. So the mapping of the 400 million short reads to the reference genome is a very computation intensive task. And if it is executed on a normal machine of 16 core, so mapping of the 85 million pair and reads to the HG38 human genome, it takes around 6 days on a 16 core machine. Here I'm talking about the 400 million. The data here I have mentioned about the 85 million. So it takes 6 days. Now you can imagine that if you have a 400 million, then how much number of days it can take. It may go to 25 days to 30 days on a 16 core machine. So it requires high performance computing or computer grid for that. Also, it has the challenges of faster mapping algorithm. Here we need a faster and faster mapping algorithm, and there are lots of algorithm which was developed, uh, like one of the method is a BWA, Burroughs-Wheeler algorithm, that was developed, but later on some other laboratory has developed, like Botai and Botai 2. And Botai 2, they claims that their algorithm is an ultra fast algorithm for the alignment. So basically we need faster algorithm for the alignment, because we have to align very large number of reads of 400 millions of reads. So we need a very faster alignment algorithm. Also, the data is of very huge size, so we may require so much memory space. So we need to manage the memory very efficiently. And we also need data compression technique, because this much data cannot be accommodated into the primary memory. So we need to basically compress the data. So, and the operations on the data must be done in the compressed file format. One of the format that I know and which is popularly used are Burroughs-Wheeler transformation. Burroughs-Wheeler transformation is one of the lossless data compression technique, and that is known as BWT, Burroughs-Wheeler transformation. BWT is applied to the genome so that the data can be compressed. So on a normal computer or a desktop computer that we have with the 4 GB of RAM, uh, we can work on that even 4 GB of RAM because the data is compressed using the BWT. So the data is now can be accommodated into just 4 GB of RAM. Otherwise, if you will not use a data compression technique, then it may require at least 64 to 128 GB of RAM. So still we have a scope to develop some further better data compression algorithm here so that the genomic data can be compressed for the purpose of storage so that it may take less storage space, and also the data transfer between the computers or from the server to the client, data transfer should also take less bandwidth. And most importantly, we need massively parallel algorithm. So we need a very parallel algorithm, and not just parallel, rather it should be massively parallel, so that uh, like hundreds of or thousands of reads may be mapped to the reference genome simultaneously. Not just one, rather thousands of reads may be aligned to the reference genome in parallel. So that may exploit the, if you have the multi-core processor or if you have high performance computing, then it can exploit those machines. In several servers, it also utilizes the GPUs today. GPUs are very cheaper, so the GPUs can be utilized for this purpose. Now, next challenge is computational challenge is molecular dynamic simulation. Molecular dynamic simulation is a kind of computer simulation about molecular dynamic simulation is like a computer simulation method for analyzing the physical movement of atoms and molecules. So the atoms and molecules are allowed to interact for a fixed period of time, giving a view of dynamic evolution of the system. And basically, this method is applied mostly in chemical physics and material science and biophysics. And it is generally utilized for the purpose of finding the binding affinity or in a in a complex between the drug and the molecule and the target protein. So it tries to find the stability, how much stable it is. And a normal MD simulation of a complex for just 100 nanosecond, it takes around 10 to 15 days on a normal, core i7 machine. So on a core i7 machine for just 100 nanosecond, it takes 10 to 15 days. If you,
Khalid sir,
yes,
sorry to disturb you, sir, but yeah, now it is fine. Actually, we were seeing only the first slide of your presentation. Now it is fine.
okay. So basically, the molecular dynamic simulation it is also very computational intensive task. If you'll do the molecular dynamic simulation for just a few nanosecond, as I have mentioned, 100 nanosecond, it takes around 10 to 15 days on a normal core i7 machine, right? So, but if you take for not nanosecond, rather if you go in a millisecond scale, then it may take a month just for a one complex, right? So if you are having 10 or 20 different complex, it may take around 3 to 6 months on a core i7 machine, right? So we need to further, there are lots of optimization scope in this field. We try to optimize our molecular dynamic simulation. And third one is Third one is molecular modeling. In the molecular modeling, we try to model some of the molecules, right? Like, for example, today, the COVID is an ongoing problem, and the researchers has already identified some of the some of the target protein. Sorry. So, in the COVID infected patient, the researchers has already identified some of the host protein, and that must be targeted by the drug. Like today, I think 4 or 5 different protein has been identified as that can be targeted with some kind of drug. So we wanted to discover or engineer some kind of molecule that should go and bind with those proteins, right? So we need to basically model such kind of molecular structure. So that modeling a molecular structure uh, is also very computational intensive task. Next is a genome assembly. Uh, Genome assembly. Before understanding the genome assembly, we should first know that our the DNA is very long in length, so the sequencing machine cannot do the sequencing of the entire DNA together. So the DNA is actually fragmented into smaller parts. It is chop up, the DNA is chop up into smaller fragments, and the smaller fragments are being sequenced independently, and they are stored into the databases, right? And they produces the reads. So uh, one fragment per reads is being produced, right? In the form of, like here in this diagram, we can see these small chunks are there. So we can assume that this is the fragments of the DNA which was sequenced, and now those sequences are now available. Now, because we have chopped it up into various fragments, and it's not just a smaller number, rather it is in millions, so millions of fragments. So basically we have to assemble all these fragments. So we can assume it like we have one straight line, and we have chopped up the straight lines into smaller fragments, and then now we have to assemble it in a proper order. So basically it is the problem of just genome assembly. We have to assemble all the reads, right? So this is also very computational intensive task, and one of the very popular algorithm that we use in genome assembly is a de Bruijn graph method. So de Bruijn graph, it's a graph based approach uh, that that needs at least it needs um, more than 100 GB of RAM. So this genome assembly cannot be done carried out on a smaller computer. It requires some server with more than 100 GB of RAM, and also genome assembly. It can take a month, around month to assemble the entire genome. If but the duration may vary from the size of the computer. If it is being done on a high performance computing with the hundreds of cores, then it can be minimized. So basically, genome assembly is also one of the tasks that needs lots of optimization, lots of data compression approach. Next is a systems biology, where we talk about the networks. We talk about the interactions between the various molecules of the human body. Like we have genes, proteins, we have RNAs, we have metabolites, right? So there are various macromolecules in the body, and uh, it is evident that the protein actually functions in the body by interacting to each other. So protein interact with other proteins to do any kind of function. So we have a protein to protein interaction network. Also, we have interaction between genes, between gene to gene, gene to protein, and interaction with the metabolites, right? So systems biology, basically, it is one of the sub area in the field of bioinformatics, where we try to study the networks in the biology. So we can say that it is a network science in biology, where we try to study uh, the interactions at a different scale. In this diagram, you can see there is an interaction at a different scale. We can start with the lower scale, that is at the gene level, then at the molecular networks. So here we can talk about only uh, we can talk about the homogeneous network. Homogeneous network means it has only genes, only single kind of entity in the network. But at a upper scale, then it can have molecular network. Here, molecular, we can have genes, proteins, metabolites, and other macromolecules can be considered. So it's a very complex network. So, and then if we'll go further at some more higher scale, the cell also, there is a cell signaling. So the cell also signals another cells. The cells are also like connected ones. So and we have different types of cells as well. So here we have a cellular network, and at a very highest level we have a organ networks. So there is also connectivity among the organs, right? So these all are about the systems biology, that we try to develop some kind of uh, a network dynamics study, some approach where the network can be constructed, because the construction of the network even at a very lower level, like at a gene level or at the molecular level, it's a very complex task. How protein interact with each other, uh, we need to develop some computational approach method that can infer all these network information. So we can call it as a network of networks. So there are several uh, thousand networks, and then that makes a complete network, right? So here we need to apply lots of network science approach that is being applied in other field of studies. We need to borrow those, and we can try those approaches here to analyze these complex networks. Now the next approach is which is a very computational intensive task, is a formulation of nanoparticles. Right? Today, everyone know that today we have nanoparticle that was discovered that has the size in a nanoscale. So now is it possible to develop the nanoparticle as a drug? Because nanoparticle is a very small in size, and that can easily go inside our body, and that can easily reach to the targets. So basically we need to do the formulation of the nanoparticle in the form of a drug. So we have to formulate it. So the computer science, especially the bioinformatics, computational biology, it is contributing a lot in carrying out the simulation study. So with the help of the simulation studies, uh, the nanoparticles may be formulated, and it can give some hypothesis about the formulation, which can later be validated in the wet lab environment. Then computer aided drug discovery, designing, and repurposing. So computer based drug discovery, designing, and repurposing, it is also a very popular area of research in the field of bioinformatics. With the help of computer algorithm, it is possible to design some drug, or to discover some drug, or even we can repurpose the existing drug for a particular disease. Like for example, for a COVID-19, when the target protein was already discovered, Then the entire community who are working on the computer-aided drug design, they started hunting for a suitable drug. They started hunting for suitable drug that can have a very good binding affinity with the target protein of the COVID-19, right? And they started searching for some of the drug from the existing library, like we have FDA approved drug library, which was already approved by the FDA. So they started looking for some of the drug from those library which can have a very good binding affinity with the host protein, right? And they came up with a very some kind of drug that was used in malaria that can have very good binding affinity. So for the timing, it can be used. So basically, in the computer-aided drug design, we have one of the approach that is known as uh, molecular docking study. Molecular docking study, where we have a library of drug and we have the target host protein, target protein which we are targeting. So what we can do, what the algorithm can do, it can take a drug from the library drug compound, it can take the structure, and it can try to fit with the target protein structure, right? So it will try to fit and it will find out some fitting score. Fitting score may be in the form of root mean square deviation or some other scoring method. So it will try to screen the entire library and try to dock with the target protein. And then we can analyze those results in the form of binding affinity and in the form of free energy and some other parameters. And based on that, we can shortlist some of the drug. Like we can shortlist of 5 drugs from those library which has very good binding affinity. So at least we have screened some of the drug that may be suitable for a particular kind of disease, right? And that for further reliability and validation, it has to go to the wet lab experiment where it can be further validated. But at least the computer aided drug designing process will help you to screen some of the suitable drug, or of either it should be it can be a new drug or maybe repurposed from the FDA approved drug, or it can be from Ayurveda, it can be from herbal medicine or from any kind of medicine. Now, one of the very interesting challenge today that today we have the concept of personalized medicine and personal genome. Today, whatever the medicines that are prescribed by the doctor, uh, that is a generic medicine. That is a general medicine for all human body, right? So everyone is going to take the same kind of medicine. While prescribing the medicine, our genomic information is not considered. Doctor never, try to scan our genomic information, because each and every person has a different genome. They carry a different genome, right? So is it possible that the medicine can be prescribed based on the genome of our body or genome of an individual? So today it is, yes, it is possible that the drug which is given to me may not be suitable for other human other body, right? So there is a concept of personalized medicine, that the medicine may be developed just for me, just for a particular person, for a particular disease, and based on the genomic information, right? So the concept of personalized medicine has given lead to the personal genome project. Personal genome project, where the individual person can go and get their sequencing done for their own genome, and the entire genome of the individual will be available in just a smart card. So you will be you will go and get your personal genome sequencing done, and that will be stored in a simple chip, right? And you can carry that particular chip. But whenever you will visit a doctor for any kind of disease, then you are supposed to carry your DNA, a personal genome as well. And the doctor will analyze your genome, and based on your genomic information, the doctor will prescribe the medicine. right? The medicine and the doses will also be, set according to that. So that has given rise to the concept of personalized medicine. And because of the personal genome project, it is available, it is feasible today. And in some of the countries, it is just in a starting phase. And today the sequencing cost has been reduced very much. So the personal genome sequencing just cost between 100 to $200, but it varies from countries to countries. So within the 10,000, or 15,000 Indian rupees, it is possible to get your sequencing, done. Even I know that in the Lal Path Lab in India, Lal Path Lab has facility for genome sequencing. So if somebody wanted, can visit and get their sequencing done, and they can carry their. But still, in India, it's not in practice. But gradually, we hope that it will be practiced in India as well. But in some of the developed countries, it is in this the initial stage even. So it is expected that there will be a paradigm shift in the treatment, where earlier we have one size fit all medicines. So the same medicine is fit to all the human body, but in some of the human body, there may be very adverse effect and zero benefit. And in some of the group, that medicine may be, beneficial, but it will not fit to every human body. So there will be a huge paradigm shift from the one size fit all medicine to a kind of personalized or precision medicine, where before prescribing the personalized medicine, the doctor will analyze your genomic information, and there are lots of analytics will be done. And then based on that analytics, the doctor will prescribe you the medicine. And even in the working model of the personalized medicine, the doctors are connected to the pharmaceutical industries, right? As soon as the doctor will analyze your genome and prescribe some medicine, uh, that information will reach to the pharmaceutical industry, and they will have very rapid way to develop the prescribed medicine. And within a day, or by the evening of the same day, uh, the medicine will be delivered at your doorstep. So this is a kind of, the working framework for this personalized medicine, right? So I mentioned some of the challenges, computational challenges in the field of bioinformatics and their working scope. Here I have listed some of the grand challenges uh, in the bioinformatics research. One of the grand challenges is developing the artificial cell, right? The human body is made up of trillions of cells. So is it possible that can we develop some simulated artificial cell? We can call it as a E cell, right? So yeah, the people has, some the researchers are actually working on development of the E cell, but they have started working on a very small genome, like some of the bacteria, which has the size of the genome is very small, right? So, like Homo sapien has 3.1 billion base pairs and around 20,000 genes. They have started working on some of the smaller organism which has less than 100 genes, right? So it is one of the grand challenge that, and the another grand challenge is development of the artificial organ, like some kind of artificial organ, engineered organ that should have the the data, that should have the entire model, simulated model should be integrated in the artificial organ. Then knowledge management is also one of the grand challenge, because the data is, very much that in a voluminous amount, and how to manage those knowledge. So we need to create a knowledge based system. One of the grand challenge is a personalized treatment. I already mentioned, so it is also one of the grand challenge. And uh, next is a early disease diagnosis. Diagnosis of some disease. Today, the cancer is diagnosed when there is some symptom which has arises in the body. But we need to detect the fatal diseases like cancer in a very early stage, maybe 10 to 20 years ahead. So we can just predict, so that necessary measures for its avoidance can be done. So it is one of the challenge to predict it as a very early stage. And then genome editing. Genome editing is possible today, but it is one of the challenge to put into the practice. Now I will few minutes I will talk about the big data problems in the bioinformatics. So we can see that the data is coming from various sources, like we have microarray data, we have gene interaction, protein-protein interaction, sequence analysis, evolutionary pathway. So these all are different kind of data analytics that we do in the field of bioinformatics. And each and every data is of very huge size, and that cannot be accommodated into a traditional file system, and that cannot be handled like a traditional in a traditional way, right? So we need to study at a different level, like we can study at a compound level, gene function, cells, and disease level. So whatever that we are analyzing, basically we need to have or handle the problem of the big data, like a storage problem. I have already mentioned that even for a Homo sapien NGS data, it can create up to 200 gigabyte of data per sample. So 200 gigabyte, for a single sample. So we have thousands and thousands of samples. So it has the storage problem. So there are lots of data compression approach that has been developed that can compress the data up to 80%. So we need to develop some more data compression approach for better storage. And another challenge is data transfer, because the data are stored into some kind of data repository, and now we have to download the data from the repository to our local server. So the data size is very voluminous one. So data transfer is one of the challenge here. Next one is the data security, because these all are the biological data, and the biological data sharing actually needs some kind of compliance of the security. So we need to secure those data for any unauthorized and illegal access. And computational power. We have already seen that many of the algorithm is very computationally intensive. So we need huge computing power. And the solution of all these all are like a cloud computing. That cloud computing is helping us to handle the big data problem. So every cloud has a silver lining. Cloud computing can solve all those problem related to storage, even computation, right? So even for a particular lab, if a particular lab is not having very huge computing power, then they can get access of the grid computing and huge computing power with the help of cloud. Or if we have a storage problem, we can buy the storage in the cloud environment. And Hadoop is, I think you must have gone through several lectures on Hadoop and big data, so I will not talk much about that. So the future prospects of the bioinformatics is the network-based approach. Like today we have a network-based approach for the treatment of disease, and that network-based approach of the treatment of disease will be like a person-centric based treatments. And big data analytics, along with the artificial intelligence in medicine, These are the approach that are redefining the bioinformatics. So basically, how the biological data can be exploited by the data analytics framework, big data analytics framework, and artificial intelligence, how it can be transformed to the practice. So from the bioinformatics, now we are living in the era of translational bioinformatics, right? So there are lots of things that are being developed in the field of bioinformatics, how this can be transformed into the clinical practices yeah, in a medical science. Right. Now, uh, I will show you some of the research contributions from my lab. We are trying to do something, in the field of bioinformatics. Although I am from computer science, but I'm trying to do something in the field of bioinformatics. So, with the help of the collaboration with the different biological lab, basically I'm able to do it because it has lots of biology. So one of the area like inference of large scale gene regulatory network from high throughput data. In this particular field, there are some of the research papers from my lab that we have developed some novel methods for the inference of large scale network. So basically it is a data driven approaches where it will take the data, large scale data as an input, and then after processing and pre-processing and a group of methods that we have applied for the construction of the large scale gene regulatory network. Then in another field that is called molecular docking for drug design and drug repurposing, we have also contributed some of the paper. One of them was in collaboration with some other lab. We have published in scientific reports. In one of the paper in collaboration with CDRI, Lucknow, we have developed some mathematical model for some kind of interaction. Then with the help of one of the collaborator from the dental department, we have proposed some drug, and we have seen the effect of the turmeric here, curcumin, like turmeric, their effect in the fibrosis. We have seen in this paper. In a nanoparticulate, also we have done some kind of simulation with the help of some lab, and we are also working on the optimization of neural network and machine learning based approaches, where we have contributed some of the paper. We have also worked in the field of COVID-19. One of the paper that in collaboration with some of the lab, uh, it is accepted in Journal of Traditional Medicine. It is in the collaboration with one of my African collaborator. They have the African medicinal plants, and they wanted to see that uh, any one of the African medicinal plant, whether they can be used or utilized for the treatment of COVID or not. So here we have screened some of the medicinal drug from the African herb. From the Indian Ayurveda herb also, in collaboration with All India Medical, we have screened some of the herb that we use uh, in our kitchen, in Indian kitchen, that is being used as a spice, that we have using some in silico study. We have proposed some, but it is still, this paper is under review. Recently I have edited one of the volume of 22 chapters in the studies of computational intelligence, and last month it was published. These are some of the references. So thank you for listening to me, and if you have any questions, then I can address. Thank you so much.
Uh, thank you, Dr. Khalid Raza. In fact, we have some questions from participants. So, should we take up these questions, Khalid Raza?
Yeah, sure.
Yeah. Okay. So, the very first question is, what is the extent to which personalized medication can contribute to better health outcome for an individual, given that family history is already a part of clinical diagnosis today?
Yes, basically, today, yes, it is in the practice that doctors generally ask for some kind of history, family history, before the doctor is going to diagnose, right? But still, that information that we are passing to the doctor, that is not very evidence based. That is only we are going to pass, yes, this particular disease was there in my family or a nearby family. But in case of the personalized medicine, here we are talking about the genomic information. Although it is evident that we have very similar kind of genome from our parents, but there are lots of mutations occur, right? So there are lots of mutation that may occur in our body. So it is not always necessary that the same genomic information that we can have from our parent. Although the genome of ours will be, very similar to our parents, but there are lots of mutations that may occur. So basically, in personalized medication, we are talking about the current genomic information rather than the just past history.
Oh, okay. So, next question is, what are the benefits when we identify the structure of protein using AI technique?
Benefits, yeah, actually, the structure of the protein is actually developed using a wet lab technique that is called X-ray crystallography. This is one of the very successful approach, wet lab approach, that takes lots of time. Maybe it may take a year also, or it needs so much of funding as well. Uh, but today we have, of course, we have the AI-based approach to model the protein structure. But again, the problem is of the reliability. So the AI-based approach is based on some algorithm, some kind of training, machine learning, or maybe some other kind of computational approach. So yes, it can be done, but still it has the problem of reliability. Still, the X-ray crystallography, that is the method which is more reliable.
Okay. The same participant has asked one more question. Rather, this is he needs, some sort of suggestion. I understood that the input of computer programs in the domain of bioinformatics is either DNA sequence or ammonia acid sequence. Is it right, sir? If yes, how can we get DNA sequence from blood sample?
Yeah, so yes, this is one of the data that the participant has mentioned. If you talk about the sequence analysis, bioinformatics is not just a sequence analysis. Bioinformatics, the sequence analysis can be one of the data, right? So the two types of data, DNA sequence or amino acid sequence, right? So that there, but there can be a different kind of data as well.
Okay, okay. There is one more question. Generally, what is the qualification of people who go into bioinformatics research? So, what should be the qualification?
Actually, bioinformatics is an interdisciplinary area, right? So, anyone with a science background can go for it. Like in my department at Jamia Millia Islamia, Department of Computer Science, we are running MSc Bioinformatics course here, and that's why I am very much indulged in teaching bioinformatics here. So any graduate, science graduate can come. Like we have a student from BCA, we have a student from botany, MSc Life Sciences, but anyone can come here. Any science graduate can go for it.
There's one more question. Can you share the open problems in bioinformatics?
Yeah, open problem we have I have already mentioned about the grand challenges
Gotcha.
at the end of my lecture, like simulating the entire cell. Is it possible to, because the entire function of the cell, whatever the function a cell is doing, can we simulate the entire cell? develop a computational version of it that is called e-cell. It is one of the open problem. And next is related to the application of personalized medicine. Is it possible to have we have a complete working system for the personalized medicine where we can connect the patient with the doctor with pharmaceutical industries and many other.
There is one more question here. Has the improvement been established in statistical terms?
Improvement in what?
Um, Shazia Parvez, are you there? Can you elaborate your question? You can unmute yourself and you can elaborate your question. Referring to personalized medication. So Khalid Raza, she actually wants to know, and she is referring to personalized medication.
Personalized medication.
Yes.
So statistically, how much it is established?
Yes.
Yeah, statistically, I cannot quantify in terms of accuracy right now because just in infancy stage and in terms of the if she is asking about the accuracy in percent, so we cannot quantify. But still, it is in infancy stage. In the USA, some of the lab has just initiated one of the pilot project on the personalized medicine. Is not quantifiable in terms of percent.
There is one more question here. What are the pre-processing techniques that are usually required for the medical image data?
Uh, I have not mentioned. I think I'm not the expert person in image processing.
Yes. Okay, we can take another question here. Professor Kiran has thanked you for your reply. And if I wish to develop a health instrument for social application, what can be the procedure?
Can you repeat the question, Dr.
If
Faisal?
if I wish to develop a health instrument for social application, what can be the procedure?
Okay. Procedure can be like today, the people are trying to exploit the IoT based approaches. They can develop some kind of sensor, and uh, sensor can be utilized for the purpose of taking the vital signs and symptoms of the body on a real time. So the sensor, we can have a band for putting in the hand, like a watch, and it can take all the vital signs from the body, and it can sync the data to the cloud. And in the cloud, we have already deployed some kind of computational analytics model, and that the data will be collected and analyzed. And if there is any kind of uh, there are any kind of irregular signs and symptoms in the body, it can generate alert as well. So in one line, we can say IoT can be utilized for developing the instruments.
One of the participants wish to use deep learning to build the software for the instrument.
Which kind of instruments related to a particular vital signs of the body, like a heart rate or pulse rate?
In future, I think that Professor Kiran will work on. So, Professor Kiran, if you are you there? You can elaborate some of your questions.
Good morning, sir. It was a privilege listening to you, and it was a wonderful presentation, sir. That is the reason I have involved, and I'm asking more questions.
Thank you so much,
Actually,
sir.
Sir, actually, you have answered my query, sir. Like I have asked you to, what is the procedure to develop a social instrument. What I said was, while preparing that instrument, I wish to use this deep learning to scan or something like that. But you have answered that better to use IoT so that this machine, whatever we wish to develop, is usable. I have the answer, sir. Thank you very much.
So I just can add yeah, one more thing to your answer. Uh,
Please.
Yeah. So today, every people they try to carry the cell phone, right? And the cell phone is connected to the internet. So whatever the device we can develop that can be connected to the cell phone through the wireless, network like Bluetooth or something. So whatever the sensors that we have in the body that may be connected with the cell phone, and the data is stored into the cell phone. And as soon as the cell phone gets connected to the internet, the data is now synced to the cloud, right? So this is, I think, one of the best way to capture the data into 24/7 basis, and the data can be utilized for the data analytics purpose.
Thank you very much, sir.
Thank you.
Thank you, Professor Kiran, for your active participation. Dr. Khalid, we have some more questions.
Okay, sure.
What are the best repository for bioinformatics research?
The best repository I mentioned is NCBI, National Center for Biotechnology Information. It has more than 30 different databases. And it's not just the source of the data, rather it is a source of the learning as well. So there are lots of initial materials which are available on NCBI. And NCBI is also one of the repository of bioinformatics data.
Okay, so we have one more question, and is advisable to combine data sets from different sources, and how can this be done as each of the data sets are compiled on a separate set of parameters?
Yeah, it is feasible to combine the heterogeneous data. Like today, the people are developing several methods for the combining the multi-omics data. In one of the uh, research paper, we have also developed some of the method uh, for the combining of different data sets. Like we have the methylation data, we have gene expression data, we have copy number variations data. So these all data are coming from data, and these all are stored into different matrices. Their values may be different. Some of the data may be numerical, some of the data may be have nominal values. So there are several approaches with the help of which we can combine the data before we are going to apply any computational technique like machine learning. So data combination is possible, but as you are the data is must have been gathered on a different parameters. So yes, we need to compromise. Otherwise, the data, if you are taking from the third party, then we will have to compromise because all the parameter was set by them, right? But at an individual level, if we are developing the data from our lab only, it's a primary data for us, then we can try to set the similar kind of parameters for the data.
The last question is, can we use sentiments of people to develop drug and medicine?
Sentiments to develop drug and medicine. Yes, sentiments can be used as one of the parameter, like the development of drug and the machine for that, like a recommender system. If you talk about the medicine recommender system, it cannot only rely on the sentiments of the people, but the sentiments may be one of the parameter beside the other parameters. Sentiment may have some participation in this.
Khalid, could you please suggest some study resources for um, of bioinformatics for beginners? That will be helpful for the
Yeah, yeah,
participants as well.
yeah. There are actually lots of titles which are available, uh, like Bioinformatics for Dummies. The dummy series of the book. I think everybody must be knowing Java dummies and Python dummies. So we have a dummy series of the bioinformatics as well that has very initial, they have mentioned about some of the fundamental aspects of the bioinformatics. We can start with that, but I will recommend that. Before you are going to learn bioinformatics directly, one should know the molecular biology, because bioinformatics, in other terms, it is known as the science of molecular biology, where we talk about DNA, RNA, and protein. So basically, if you someone start from the molecular biology and try to understand the central dogma and how the DNA replicates, how it is transcribed and translated to function protein, all this, then I think the people can easily understand the field of bioinformatics. And
Thank you. Thank you.
there are some of the research papers that was written by the computer scientists, like molecular biology for computer scientists. So one of the research paper that is very good, I also started with that, and that molecular biology content was written by a computer scientist. So a computer scientist can easily understand those terminologies.
Thank you very much, Dr. Khalid Raza. It was very useful session, in fact, for all of us. And participant has also appreciated your effort, and you can see their appreciation on this chat box. And thankful for your all wonderful presentation. And we wish in future we will meet in physical mode as well. Thank you so much, Khalid. Khalid.
Thank you. Thank you for inviting me, and thank you all the participants for listening me and having patience. Thank you so much.
The participants may leave this session, and we will again meet at 12. So please join the next session at 12. Thank you so much.